I am a medical oncologist, as Lynn mentioned. I'm new to Emory University. I was up in Boston for 7 years before that at the Dana-Farber Cancer Institute. I spend about 25% of my time caring for people with advanced cancer. So that's what I'm going to be speaking about today. The disclosure that I wanted to mention up front is that I actually am not a lung cancer specialist in terms of the types of folks I care for. I'm actually a prostate cancer doctor. So when I'm in clinic, I'm mostly caring for men with prostate cancer. However, the research that I do really spans several different cancer types, and we've done some what I think is really exciting work in the lung cancer space with some fantastic collaborators. So just wanted to give that disclosure. So, happy to answer questions, put them in the chat, interrupt me, whatever, if questions come up as I'm speaking, but did just want to give that caveat. Today, I'll be speaking about lung cancers that change from non-small cell to small cell lung cancer. And the more detailed title is Detecting Small Cell Transformation in Patients with EGFR Mutant Lung Adenocarcinoma Through Plasma Epigenomic Profiling. And that sounds like a mouthful, but I'm going to do my best to explain what we've done in the next little bit. So I sort of mentioned the disclosure. This is a slide that's probably not necessary to the folks on this call, but I think a really important and sobering piece of data that puts the importance of this work into context, which is really that whether you're a male or a female in the United States of America, that lung cancer has been, for the last 80 years, the leading cause of cancer-related deaths, and that's for men and women, and I think really encouraging to see those red lines coming down over the last, well, I guess that was from the 90s up to 2020. And with that sort of important context, a little more background on lung cancer that's relevant to what I'm going to be speaking about today. So that small pie chart on the top left shows the breakdown of the different types of lung cancer. Lung cancer is not one disease. Lung cancer is actually several diseases. And the way that we have classified lung cancers into subtypes for decades is basically how they look under the microscope. And that there are several different tumor types that we call adenocarcinoma, squamous cell, large cell, small cell, lung cancer. And really, those names come from how the tumors look under the microscope. I'm happy to share the slides. As you can see, lung adenocarcinoma is the most common subtype of lung cancer. It accounts for about 40% of lung cancers. Small cell is one of the less common ones, about 15%. And just sort of, I think you'll see that those subtypes are relevant because really what we're going to be talking about today is largely how some lung cancers switch from a adenocarcinoma to a small cell. And so you can see that small cell is its own type that can exist when people are diagnosed with lung cancer. But here we're going to be talking about tumors that switch from one type to another. Within lung adenocarcinoma, we know that research over the last 20, 30 years has identified what we call drivers, genes that we call drivers of the cancer. So mutations in these genes lead to unchecked growth of lung cells that then turn into cancerous cells and then drives the cells to grow and spread. And so, we've characterized a lot of these genes in which mutations drive lung cancer. And one of the most common ones is this gene EGFR. It accounts for about a quarter of lung adenocarcinomas will have an EGFR mutation. And these EGFR mutant lung adenocarcinomas are really, as the title of the slide suggests, is the paradigm for precision oncology. What do I mean by that? Precision oncology is really sort of the dream of that we've had for many decades of being able to study an individual person's tumor, learn what's driving the tumor, and then target that with a targeted therapy. So we now have several targeted therapies against EGFR mutations, and this is the NCCN guidelines. It's really sort of the best resource that tells oncologists how to best treat their patients. And as you can see here, if an EGFR mutation is found at diagnosis, the first treatment are EGFR targeted therapies. And so, I really wanted to drive home that there's been tremendous progress in understanding the mutations that drive certain lung cancers. However, the sobering reality is that even these targeted therapies don't work forever. Resistance or progression is basically universal. We have these amazing success stories where people will be 5, 10 years on treatment, but the more common scenario is within a couple years, people will progress on these targeted therapies. And so understanding how the tumors are evolving to become resistant to these targeted therapies is really important so that we can figure out how to overcome that resistance and develop more effective therapies. But most of these resistance mechanisms are about half of them are the EGFR gene becoming more mutated to overcome resistance. There's other genes that are altered or mutated to become resistant. But what I'm going to focus on today is this about 10 to 15% of tumors that transform from an adenocarcinoma to a small cell lung cancer as a way that the cancers become resistant. So, the EGFR mutant lung adenocarcinoma started on treatment, and then, I'm sort of describing a common scenario, and then a couple years later, scans show new spots somewhere in the body, and that one of those new metastatic sites is biopsied, and the tumor is transformed from a adenocarcinoma or a non-small cell to a small cell lung cancer. So far, I've been talking about EGFR mutant lung adenocarcinoma. That's the most common scenario in which we see this transformation to small cell lung cancer. However, less commonly, we do see other adenocarcinomas with other mutations like ALK fusions or rearrangements, ROS1 fusions, rearrangements, even squamous cell lung cancers we've seen transform to small cell. So I'm going to mostly be focusing on the EGFR mutant lung adenocarcinomas that transform because that's the most common scenario. However, I do think it's important to acknowledge that this happens in other contexts, and so the implications are broader than just one lung cancer subtype. Conversation is, why do we care about small cell transformation? Sort of what are the implications of this happening where a adenocarcinoma transforms to a small cell? The first is that it seems to be increasing in prevalence, finding it more often. This is probably a result of our drugs getting better and better. If with better and better, more potent drugs targeting EGFR signaling, the cancer cells are desperate to figure out ways to survive. And so, I think we're seeing that more commonly cancers are transforming from an adenocarcinoma to a small cell. Unfortunately, we know that when tumors transform to small cell, that's associated with worse outcomes. Small cell lung cancer tends to be among the most aggressive of the lung cancer subtypes, and tends to not have quite as good treatment options. However, there are some treatments for small cell lung cancer that they're just saying we treat small cell lung cancer different than we treat lung adenocarcinoma. There's been tremendous progress such that we really tailor the treatment that we give to the specific tumor type and tumor subtype. And so knowing that someone's transformed to small cell lung cancer, the treatment's going to look quite different in terms of the chemotherapy regimen, the immunotherapy, potentially targeted therapies than we would give with lung adenocarcinoma. So really knowing that the small cell transformations happened allows us to better and more accurately treat people. And then I think, perhaps most importantly is that there's incredible interest and efforts going on in the lung cancer research community to develop better treatment options for either small cell diagnosed upfront or transform small cell, with lots of clinical trials ongoing. And so, really, I hope that drives home why we care about this. And as a result, the NCCN guidelines are very clear. This is a quote from the guidelines that say, for any patient progressing on targeted therapy, histologic transformation, such as small cell, is a possible mechanism of resistance. Thus, tissue biopsy of a progressing lesion should be considered to evaluate the tumor to see if small cell transformations happen. So the guidelines recommend looking for small cell transformation. However, there are major limitations in flaws in how we currently go about finding this small cell transformation. As I mentioned, all of these classifications of these tumor subtypes are based on looking at the tumor under a microscope. And to do that, you need tumor tissue to look at under the microscope. And so really the current paradigm, as the guidelines mention, is to get a biopsy. And as folks on the call who've gone through that know, biopsies can be invasive and painful and have lots of risks. And as a result, can really only be performed once, maybe twice. And so, challenges with biopsies. And the reality is that they're hard to do sometimes, and whether it's for safety concerns or resources, or, we know that less than half of patients get a biopsy after progressing on their targeted treatment. And so, I think that tells you right there, we're probably with this current approach missing this more than half the time because we need a biopsy to find this small cell transformation, and more than half of people aren't getting biopsies. And then the last piece is actually a really important one, which is that this idea that of tumor heterogeneity. And what I mean by that is that even within a single individual, there can be different tumor types. And so, this small cell can emerge, for example, in a liver metastasis, and but not be in the primary lung or the primary lung cancer. And so, if you biopsy one site, you might actually miss the small cell transformation. That's what we call spatial heterogeneity, as shown on the right. And then on the bottom is what I mentioned, is usually people can only get one biopsy in their disease course, and this small cell transformation can happen at any time. And so, if you got a tumor biopsy after the first progression, and the small cell transformation happened later on, you might not detect it. And so, really, pretty major limitations of our current approach, highlighting the need for better tools to find small cell transformation in the clinic. So this is where liquid biopsies come in, and this is what my research focuses on. So liquid biopsies are based on the observation from almost 100 years ago that tumor cells actually shed DNA into our bloodstream. So when we have tumor cells, we can actually take a blood sample, just a routine blood draw, like folks get in the clinic every day, and actually study the tumor DNA that's circulating in the bloodstream. And we call that a liquid biopsy. As you can imagine, this has a lot of potential value over doing a traditional tumor biopsy. First is that it's easy to do. We can do it repeatedly. You could do it every couple weeks, every couple months, as opposed to a tumor biopsy, which really, as I mentioned, you can only do once or twice. It's a lot less invasive for folks, so more patient friendly. And that issue of heterogeneity, this idea that there's multiple tumors with multiple different features in the body, if all those are shedding into the bloodstream with a single blood draw, we may be able to sort of sample information from all the tumors in someone's body. And so this liquid biopsy approach really has incredible power. And liquid biopsies are already transforming cancer care really across the disease spectrum currently. So now going even beyond lung cancer, just to sort of cancer care at large, we're using liquid biopsies for early cancer detection, to detect the presence of residual cancer after surgery, molecular profiling, which is sort of what I've been talking about, finding specific tumor features that we can target with targeted therapy, and then using it to monitor to see if a certain treatment is working. And so all of these, there are several examples I could highlight where we're using liquid biopsy for these in the clinic today. For the molecular profiling, I'm going to highlight that because there's almost 20 or so FDA-approved liquid biopsy tests to look for these tumor mutations that we have targeted therapies for. So I have a list of those on the right here. And what's particularly relevant for this group is that actually the majority of those are in lung cancer. You can see we have liquid biopsy tools to detect ALK rearrangements, EGFR mutations, HER2 mutations, KRAS mutations, MET, ROS. And so really the liquid biopsies are already part of standard of care for many people with lung cancer. However, there are limitations of these current liquid biopsy approaches that I'm going to speak to in a little bit, for why we've sort of developed and used a new approach to detect small cell transformation. So it's highlighted here, and this is where part of this talk by necessity is going to be a little technical, but certainly feel free to interrupt with questions. So I've been talking a lot about genomic alterations, and genomic alterations are mutations or changes to the DNA within a cell that then drive cancer. We've actually found that genomic alterations aren't really different between small cell tumors that transform to small cell and tumors that stay adenocarcinomas. And what's really different is actually the epigenomic profiles. Don't worry if that's a foreign term, I'm going to talk about epigenetics or epigenomics in a moment. But just to say that sort of the traditional genomic analysis isn't going to allow us to find these tumors that transform to small cell lung cancer. And we need to look at a different type of tumor feature called epigenetic or epigenomic changes. And what my research over the last 7, 8 years has focused on is developing liquid biopsy tools that enable us to study tumor epigenomic features in the bloodstream to be able to use liquid biopsy to detect things like small cell transformation. This was about 3 years in the making that was just published in September, a couple months ago, was can we use these new liquid biopsy tools focused on tumor epigenomic profiling to detect small cell transformation. And this is the publication. I'd be happy to share this with Lynn, and she can circulate it to the group that I'm going to be speaking about today. As you can see, just came out on September 3rd, so fairly hot off the press. So a little background on epigenetics. Epigenetics is the study of how cells turn certain genes on and off. The example that I use is that every cell in our body has exactly the same DNA. So how does a heart cell become a heart cell and a brain cell become a brain cell? Epigenetics is the answer. If every cell has the exact same DNA and genes, the way that a cell becomes what it is a very coordinated process by which certain genes are turned on and certain genes are turned off. And that's different in every cell in our body. And epigenetics is how the cell turns those genes on and off in a coordinated fashion to make a heart cell, a brain cell, a brain cell. And it's what makes a lung adenocarcinoma cell an adenocarcinoma cell, and a small cell a small cell. And so, with that context, for a cancer cell to go from an adenocarcinoma cell to a small cell lung cancer, it has to completely reprogram its epigenome or its epigenetic profile for this to happen. And so our approach was to leverage the fact that these tumors undergo this full-scale reprogramming of their epigenetic state to try to develop an epigenetic signature that allows us to detect this transformation from one cancer subtype to another cancer subtype. So I think a picture's worth 1,000 words. So here is an example of two tumors, a lung adenocarcinoma and a small cell lung cancer, one that started as a small cell. And without needing to go into too much detail, suffice to say that we profiled several epigenetic features in these two tumors. And some of them are epigenetic marks that mark active genes. Some of them are marks that mark genes that are turned off. But all that to say is you can see by eye that the epigenetic profiles of these tumors are opposite, right? They're totally different. And this is at a gene that we know to be active in small cell lung cancer. And so the epigenetic marks of active genes are present in the small cell lung cancer and absent in the non-small cell. And then this black mark is one that marks genes that are turned off. And again, that's there in the adenocarcinoma, and that's not there in the de novo small cell. And now, sort of the really neat thing is this third column here is the profile of a transformed small cell lung cancer. So this is a tumor that started as an adenocarcinoma and transformed to a small cell. And what's really striking, this tumor that's transformed from an adenocarcinoma to a small cell lung cancer, you can see its epigenetic profile has transformed as well to really sort of, no longer resembles an adenocarcinoma, really resembles a small cell. And so, as I've mentioned, we sought to leverage these epigenetic profiles or develop signatures that allow us to track the presence of these different tumor types. And so here's a schema of our study. I think it's sort of a nice pictorial representation. I'm not going to go through it in too much detail here, but we'll sort of use it to describe at a high level what we did. So the first thing we wanted to do is really comprehensively profile the epigenetics of these different cancer types. And so working with our colleagues at the Mass General Hospital Cancer Center, Aaron Hatta and Catherine Metter, Catherine was one of the first authors on this paper, we obtained lung cancer tumors that were lung adenocarcinomas de novo small cell lung cancer, so tumors that started as small cell lung cancer, and then a set of tumors that transformed from lung adenocarcinoma to small cell. And what we did is we performed really broad and deep epigenetic profiling of those tumors. And we used that information to identify signatures, epigenetic signatures that distinguished the different tumor types. These graphs are a little complicated, but essentially what we found first is that really, what this data shows is that the transformed small cell lung cancer tumors, for all intents and purposes, have epigenetic profiles that really look like the de novo small cell. You can see for example, let's look this plot right here, where you can see the lung adenocarcinoma tumors are over here, and the transformed small cell tumors have really completely shifted from here this way to sort of cluster with the de novo small cell. So again, just more data to support that the epigenetic state is really transforming as the tumors transform. And then what we sought to do is identify sites across the genome that were highly different between the small cell and the adenocarcinoma. And that's what this shows. And what, each row here is a site, and these are all different tumors. And what we found is that we could identify more than 5,000 sites that were epigenetic sites that were sort of enriched in the small cell tumors relative to the lung adenocarcinoma tumors, and more than 5,000 sites that were enriched in the lung adenocarcinoma tumors compared to the small cell. And so we use that information to essentially build a signature based on those 10,000 or so sites to distinguish the two different tumor types. So that's sort of background. And then here's where we get to the liquid biopsy piece. So what we did is we actually, again, with our colleagues at MGH, we collected plasma samples, blood samples from patients with metastatic lung cancer who had either EGFR mutant lung adenocarcinoma or patients whose tumors had transformed to small cell lung cancer. We used some new methods that we developed to profile the epigenetic features of the tumor DNA in the bloodstream, and then ask the question, how accurately could we take a blood sample from someone with lung cancer and tell whether they had EGFR mutant lung adenocarcinoma or had transformed to small cell lung cancer. And really excitingly, using that data from the tumors that we had generated, this is now looking at, we generated a model that we called a small cell lung cancer risk score, and we applied that to the blood samples from patients with EGFR mutant lung adenocarcinoma and small cell lung cancer, transformed small cell lung cancer. And what we saw is that the small cell lung cancer risk scores were a lot higher in patients with transformed small cell lung cancer, as we would hypothesize or expect or hope to see. And then, so that was really encouraging. And then we used a method called ROC on the right, but basically it's a method to look at how accurate the test is. So how accurate is it for discriminating EGFR mutant lung adenocarcinoma patients from patients with transformed small cell lung cancer. And what I'd highlight here is the AUROC of 0.94, which in plain English means this test had 94% accuracy for telling whether a blood sample was from a patient with EGFR mutant lung adenocarcinoma or a patient with transformed small cell lung cancer. And in the world of sort of developing diagnostic tests, take my word for it, that's pretty good. So we were really pleased with the accuracy that we were able to see in this cohort. But I think this is probably, I think the most exciting is sort of the application of this for individual people. Now, this is not someone who was able to benefit from this test, but I think this was a patient, or we have two patients where we had several blood samples collected over the course of their disease treatment, or the treatment for their disease, and we can show that this test, through monitoring the blood, can capture the emergence of this small cell transformation. And so walking through on the y-axis, sort of here is the small cell risk score that I showed on the previous slide, where a higher score indicates a higher likelihood of a patient having undergone the small cell transformation. So this patient was diagnosed and started on erlotinib, and had a great response. They were still responding almost 3.5 years later. However, at the time of progression, there was new spots on scans. They had a blood test that actually showed a mutation in the EGFR, a common resistance mutation. And so at the time when we found that, we were switching from erlotinib, a first generation EGFR inhibitor, to osimertinib, a third generation. And so they got appropriate, second line treatment. But unfortunately, within about 9 months after starting osimertinib, their scan showed new liver metastases, as shown here. At that time, the patient underwent a biopsy of that liver metastasis, and it showed transformation from adenocarcinoma to small cell lung cancer, which is shown here. Looking at the cell-free DNA or the blood at those different time points, the small cell lung cancer risk score was low when they had the adenocarcinoma. And at the time of diagnosis of small cell transformation, you can see that the small cell risk score had jumped up, and suggesting that using this blood test, we were able to detect the presence of the small cell transformation. So this was a patient who was more recently diagnosed with EGFR mutant lung adenocarcinoma. This patient was actually started on osimertinib, which became the standard treatment when this patient was diagnosed. And unfortunately, within only a little over a year, had new liver metastases seen, and similar story, that was biopsied and showed small cell lung cancer. What I think is really, and again, we see sort of a similar trend where profiling the blood from this patient preceding the small cell transformation, you can see actually that the signal was rising. And what I think is really neat about this anecdote is that you could see the small cell lung cancer risk score sort of rising between these first two time points, and then sort of further rising between the second and third time point. But what I think is neat here is that the blood sample was suggesting the presence of this small cell transformation about 4 months before it was actually diagnosed clinically with a biopsy. And so could it have been that, this blood sample went up and this person could have gotten a biopsy several months earlier? In that 4-month interval between this small cell risk score going up and the patient being diagnosed, they were getting likely ineffective therapy. I think probably that small cell, or the osimertinib, the EGFR targeted therapy, certainly wasn't active against the small cell component of the disease. And so that's 4 months missed opportunity to potentially have been delivering more effective therapy earlier. And so I think that this sort of shows how we can use this blood-based approach to monitor people serially over the course of their disease, to see these dynamic changes emerge over time. And I should say, I hope that what this leads to is the ability to detect small cell transformation early, not continue to give ineffective therapy against the EGFR mutation, and switch over to a small cell directed treatment regimen to try to target that disease directly. And I think a lot of us are really hopeful in the field that this will lead to better outcomes for people, sort of earlier initiation of more effective therapy. And the other thing that I think is really exciting is to develop new and better treatments for different tumor subtypes. The first thing we have to do is be able to find them, to be able to study them. And so, I really think, going back to that statistic that less than half of people, in reality, it's probably more like 20%, 30% of people are getting biopsies. We're really just missing this. And, I'm hopeful that not only can this help us find these people in clinic to immediately start delivering small cell directed treatment when necessary, but also be able to find these people for clinical trials to really study and advance new therapies to really make a difference for people with small cell transformation. I'm going to wrap up and then wanted to leave plenty of time for questions. But just to summarize what I've presented is that transformation from non-small cell lung cancer to small cell lung cancer is really now a well-characterized mechanism of resistance to targeted therapy, whether that's EGFR targeted therapy in patients with EGFR mutations, or less commonly other lung cancer subtypes. And the current approach to finding the small cell transformation in clinic is really limited. What we showed in our recent publication, and I sort of highlighted today, is that small cell lung cancer has a very distinct epigenetic profile, and we were able to leverage this observation and new tools that allow us to profile epigenetic features in the bloodstream to develop a liquid biopsy to detect this small cell transformation in patients with EGFR mutant lung adenocarcinoma. So this was all done in an academic lab, a really wonderful academic collaboration with one of my former mentors, Matt Friedman, Catherine Metter, Aaron Hatta. I'll acknowledge all them in a moment. But what's really exciting is that I want to see this translated to the clinic, that we can be that providers can be ordering this for patients in clinic, finding this in clinic with a routine blood draw, and making treatment decisions to better treat their patients. And what's really exciting to me is that several companies, including some of the sort of household liquid biopsy names like Guardant Health, are actively working on taking the data that we've published and incorporating that into their commercially available liquid biopsy tests. There's very active efforts from several of the big commercial liquid biopsy companies to be able to add this feature to their products. And I really think I wrote the next few years, optimistically, I think within the next 12 to 18 months, we will see some of the liquid biopsy companies that with assays that we can order in the clinic today being able to do this type of monitoring. So I'm going to acknowledge, I mentioned Matt Friedman. Talal El-Zarif is a really talented physician scientist who led this work along with Catherine. Really generous support from the Dana-Farber, Harvard Cancer Center Lung Cancer Spore, the Lung Strong team, as well as Team 3G from the PanMass Challenge, all of whom supported this work, and really without whom the work wouldn't have been possible.